Okay, good afternoon, everyone. Welcome back and congratulations. You made it to the final lecture of the semester. You are almost done. So you all know at this point, because I keep saying it, that your final exam is next week. It is, in fact, Tuesday, May 12th. So on Tuesday of next week, please log on to the class marker website. You will have between 7 a.m. and midnight. So you basically have all day on Tuesday to log in and do the exam. When you start the exam, you will have a 2-hour window of time to complete it, okay? All right, so do you have any questions at this time in terms of how the semester is going to end next week? Okay, not
Is
hearing
this
any.
our last meeting?
Today is our last meeting. We will not be meeting again after today. So next Tuesday, after you finish your final exam, you are finished in the course.
Wow. Okay.
Okay, anything else? Okay, so certainly I will be monitoring email up through the end of the final exam period. So certainly if something comes up, send me an email and I'll try to help you as best as I can. Okay, so let's continue our conversation from last time. We were talking about a practical example of using microarrays to measure the differences in gene expression between two different conditions, such as a disease state versus a healthy state. And the specific example I was talking about is the fibromyalgia condition that many folks unfortunately suffer with across America. So, okay, I'm going to go back to that document. Okay. And let's see here and bring up the chat window so that you can get my attention if you need it. Okay. All right. So this is a piece of last class's notebook. I didn't put everything in here, but the practical workflow, which is in last meeting's notes, is here, and I'm going to continue off of that. There's something kind of interesting, or, well, it was an oddity to me because I was reading something yesterday that kind of caught me off guard. And so up here at number 4, where I gave you the historical note about the development of microarrays at Stanford in 1995, okay, the person that is typically given credit for developing the microarrays is a person named Patrick Brown, okay? And he is and actually was, past tense, a biochemistry professor at Stanford University. Okay, and so what I wanted to do is show you something kind of weird here before we get back into the content. Okay, this is a picture of Patrick Brown. You see on your screen in the upper right corner, that's the gentleman that originally created the DNA microarray that you can see highlighted here, DNA microarrays. He created them and published them in 1995. So if you're curious, he's famous enough that you can actually get a Wikipedia article on him. But I was reading something yesterday, totally unrelated to this topic, because I had an interest in something. And let me ask you, let me go back down to the chat window. Let me ask you something in the chat window. Okay. Have any of you tried the Impossible Burger at Burger King? And it's totally okay if you haven't. Okay. I personally have not tried it, and I'm not sure I am going to try it. But have you at least heard of this meat substitute burger? Okay, Caitlin, thank you. This meat substitute burger, which is based on, plant material, and it's called the Impossible Burger or Impossible Meat. Well, guess what, folks? The gentleman that created microarrays is the same person that founded the Impossible Meat Company. Okay, so up here, take a look on his Wikipedia, Impossible Foods. Right there it is. Okay, so the same gentleman that created the DNA microarrays and became famous in 1995 for that actually left Stanford about, I don't know, 10 years ago or something, because he was curious about this possibility of creating a meat substitute. And he actually is the chief executive and founder of Impossible Foods. So you have a biochemistry professor that's probably now worth about $1 billion. Okay. So it goes to show you that not all scientists remain poor. Okay. So I just thought that was a really odd coincidence that the person that made microarrays is also the founder of Impossible Foods. So, okay. Thoughts, questions? Okay. All right. So let's continue here. We're talking about this microarray example, okay, where we are trying to measure the gene expression in fibromyalgia patients and measure the same gene expression in healthy patients and measure their difference, right? Their difference in gene expression. Okay, and I mentioned last time, depending on where you set your p-value for rejection of the null hypothesis, you can get way too many transcripts that are practically useful to do anything with. Okay, it's just logistically impossible. So you have to make your p-value cutoff more stringent. And so when I took it down to 0.005, we obtained 540 differentially expressed transcripts. And I showed you this and shared this with you in the email from Tuesday's class. So hopefully you all received this, okay? So once again, what we're looking at is the measurement of genes in column B in the fibromyalgia state, the measurement of genes in the healthy state, and the difference, right? 14 minus 5 is 9, the difference in gene expression between those two states. And you get a list of 540 genes whose p-value was low enough that we could justifiably reject the null hypothesis that there is no difference between fibromyalgia and healthy patients and say that there probably is a difference with these subsets of genes. Okay, that's where we're at with our thinking. So any questions at this time? Okay, all right. So the next step is really really getting to the question that I sent to you at the end of your email from Tuesday's class. I had a question to ponder, and I forget exactly how I worded that, but the basic idea is this. Out of this list of 540 different genes, if you will, they're really transcripts, not necessarily genes, but at any rate, out of this list of 540 differentially expressed genes, which one of them or which subset of them are the real driver of the disease state? I mean, not all of these genes. I mean, it seems very unreasonable to think that all of these 540 genes are coordinating in some mass murder idea to create the disease, okay? So how do we determine what the actual driver is of the disease state? That is a very difficult thing to deal with. Okay, so let's see here. Number 7, okay? Number 7, well, let's see, I guess letter E was probably not indented properly. Okay, number 7. Once you obtain a list of differentially expressed genes, how do you know which subset of those genes are the real drivers of the disease state? Okay. Well, let me tell you a few things here. When scientists started doing these experiments, like most things, scientists are people too, right? So sometimes they make naive assumptions about things. We all do that. It's just life. It's just being human. And so when these experiments were first done, it was assumed that if we got a list of differently expressed genes, that whatever genes were most differently expressed, well, those were probably the ones that were creating the disease. And long story short, a lot of research has now gone in to prove that that's really very rarely the case at all, that just isn't true. Okay, it's not the most differently expressed gene that's causing the disease. And so it really becomes a problem. So point 7 there, how do you obtain a list of genes, a subset of the genes that are the real drivers of the disease state? So here's a philosophical analogy to think about. Okay, because if anything, the experiments we're doing them so that we can understand things, so that hopefully someday we can do something therapeutically useful to help people. So we got to try to figure out the answer to this question. And it's not an easy one to answer, and nobody has really completely answered it even today. But let me ask you something, okay? And this is going to be a little weird, but if you were an alien, looking down on a road inside Philadelphia or any other city, and you saw this rectangular object that was colored yellow moving along a piece of gravel, what would you think that object is? If you...
School bus.
Okay, thank you, Hayden. That's exactly what I'm trying to get you to see. Okay, if you were an alien observing a yellow rectangular object moving along a piece of gravel, what would you think? All right, so folks, Hayden said school bus. Now let me ask you, if you're an alien, do you necessarily know anything about school buses on Earth? Is there any reason to think that that alien would believe that that is a school bus? Do you see my point? That the only reason, Hayden, you answered school bus is because you're not an alien. You're a human being with a whole context of living your life in a society where a yellow rectangular object moving along a piece of gravel to a human being is a school bus. But is there any realistic expectation that an alien would come to that same conclusion? Do you see my point? Okay. Well, okay. So we got to be very careful about, first off, do you see the implicit assumptions we all make in our reasoning? Okay, so I did a little gotcha on that, Hayden, right? I gotcha because you said school bus, and that's what I anticipated you would say. Okay, but there's no reason to expect an alien would think it's a school bus. To an alien, it's a rectangular yellow object that's moving from point A to point B, but we have no idea what it really is or what its purpose is. Does that make sense? So let me break this down a little bit, okay? And so what I'm suggesting to you is that from the perspective of a human being looking at what's happening with cells, we are kind of aliens. We truly don't know what it is we're looking at. We truly don't know what it is we're looking at. Okay, and so we have to figure out what it is we're looking at by doing experimentation and doing a lot of logical mathematical reasoning, because that's the foundation of science. Okay, so, okay, if if we if we as aliens determine that this is some type of school bus, okay, my next question is this: how do you identify the driver of the school bus from the passengers in the school bus? How do you identify the driver in the school bus from the passengers in the school bus? What do you think, folks? Okay, so go ahead and go back into your human thinking mode. How do you identify a school bus driver? I mean, you see what we take for granted, and you think you understand the assumptions we're immediately making here? You as a human, maybe you rode the school bus to school for 12 years or whatever it was. How do we humans, whether this is a school bus or a tour bus taking a group of people to New York City for a bus tour, how do we humans identify the driver as someone different from the passengers? How do we do this? I'm not asking you how you will do it for the disease that I'm talking about, but just in everyday life right now, how do we do it?
Well, the driver's in the driver's seat
How do you know it's the driver's seat?
because there's a wheel.
Okay, there's a wheel, there's a steering column, right? All right. Okay, so fair enough. Fair enough. So, folks, does it make sense that if you have a rectangular yellow object moving from point A to point B, unless you already know who the driver is or know what to look for that distinguishes the driver from the passengers, we're going to really have a hard time identifying a subset of differentially expressed genes that actually drive the bus, that actually drive the disease process. Okay, so as humans, we identify the bus driver as the person sitting in front of the steering wheel. Okay, but do you see that when there's a group of people sitting in a bus and the bus goes from Lancaster to Philadelphia, that there's really only one person driving that bus and the other 30 people sitting behind that person are just passengers? In other words, the passengers are not responsible for physically moving the bus. The passengers are not physically responsible for creating the disease. The passengers simply go wherever the driver moved the bus. The passengers simply go along for the ride. So, in this list of 540 differentially expressed genes, what I'm suggesting to you is that we think at this point that probably most of these genes that are differently expressed are simply passengers on a school bus. They're not actually driving the disease process, but they happen to be changing because they are in some way tangentially seated as a passenger in this disease phenotype. They're somehow seated tangentially as a passenger that goes along for the ride as the true bus driver, as the true disease-causing genes do what they do. Okay. Am I making sense or have I lost you?
That makes sense. So, do scientists like clone and manipulate one gene out of all 500 and then just see like if the other ones change too, depending on that gene? Like, how do they test that?
Yeah, that's a good question, Hayden. And practically speaking, that can be done and often is done, but you appreciate if you have 540 of those genes to do, that's going to take a very, very long time, and it's going to be incredibly laborious. And that's why when we originally were up here in our notes and saying that we had 3,387 genes to deal with, that would have been even worse. So you reach a point where the number of genes that you have to investigate just becomes unreasonable and you can't do it. I mean, it would take too much time, too much man or woman power, too much money. It just, nobody's gonna do this. And that's why I get back to the philosophical analogy is that we need a logical way of trying to identify the driver on the bus when we don't necessarily have the ability to identify the driver as a bus driver sitting in front of a steering column. There is no steering column in the cell that we can look at and say, well, that's clearly the driver. And this is the problem that we have. Okay, so let me see here. Okay, so within cells, there is no easy way to identify the driver of the disease. But we do know that most differentially expressed genes are really just passengers going along for the ride. They are not true drivers of the disease. Okay, I mean, let's face it, you know, when you drive, when you were 5 and mom and dad drove you all around to, you know, your baseball games or your ballet or whatever it was, you just went along for the ride. You didn't actually move the car. It was mom or dad moving the car, right? So you simply relocated your body because someone else was driving it. In that list of differentially expressed genes, there's going to be many genes that happen to be relocated, not because they're truly driving the disease process, but because they are somehow tangentially related, as though you are related to your mom or dad. And when your mom and dad drives, you go along for the ride, okay? So I'm sorry to be pedantic, but are you with me right now? Does this make sense? Okay.
Yes.
Okay, thank you. All right, so my point is that it is really, really hard to determine the driver of a disease from a large list of differentially expressed genes. And scientists and mathematicians working in concert have been trying for a very long time to develop mathematical logic and formulas and ideas that would help with this process. And so what I want to share with you now is a way to approach this that does not always work, but has had some amount of success. Okay, so let's see here. So how do you know which subset of those genes are the real drivers of the disease state? We got into this philosophical analogy, okay? So one way to answer question 7, I guess I'll just make it bullet point 8 at this point. One way to answer question 7 from your notes is to calculate, and it's a calculation that has to be done on the computer because it involves so much volume of data, you will not be able to do it yourself, is to calculate something known as the differential. Okay, so up here, I talked about differently expressed, right? Well, that's useful. That doesn't get us the whole way. That's not how we identify the driver of the disease, okay? What we want to get at is to calculate a related idea called differential correlation. Okay, differential correlation. And so what I'm going to do is just kind of show you in a very rough, cartoonish, schematic way how that would be achieved. So Okay. I hope you are, um, let me type a question into the chat window. Have you encountered the concept of Pearson's correlation coefficient in your coursework? Do you know what the Pearson's correlation coefficient is? Okay, John. Okay, so, okay. A correlation coefficient is a mathematical way to describe if two values, like two genes, change in a consistent way. In other words, two genes would be correlated if, when one gene changes, the second gene also changes. Okay? So if one gene changes and the second gene also changes in the same in a predictable way with the first gene, then those two genes are correlated. Does that make sense? Okay, thank you, John. All right, so what you do here, and I'm not gonna get into the heavy dutiness of this because we would need like a 2 or 3 hour Mathematica session, is what you do is, okay, so over here, okay, over here, you should see an Excel spreadsheet, and I'm highlighting columns A through H as information about the fibromyalgia disease. So what you would do is take your list of differentially expressed genes, and you remember from the spreadsheet that there are 540 of them. So I'd have a list of 540 genes going down column A, and I would have their gene expression value, how abundant the RNA was in the cell of a fibromyalgia patient, and I would have the same genes going across the row, and then I would calculate in a pairwise fashion the correlation for each pairing of gene. So the correlation of gene 1 compared to gene 1 should be a perfect correlation of 1, right? If it's the same gene and I'm comparing it to itself, there should be a perfect correlation, which is a correlation of 1. That's why for gene 1 and gene 1, you have 1. For gene 2 and gene 2, you have 1. So you have this diagonal of 1s that goes down your matrix. And so the thing of it is, though, you would calculate the correlation of gene 1 and gene 2 and get an answer here, the orange answer. You'd calculate the correlation of gene 1 and gene 3 and get the green answer. And so you would calculate a correlation between every gene and every other gene in the same disease state, fibromyalgia. And you would get a matrix of all these correlations. Are you with me? Okay. All right. So you would develop this matrix of correlations, all right? And you would do that for all the genes in the fibromyalgia state. And then you would do exactly the same thing for all the gene measurements in the healthy condition. Okay, so you would develop all these correlation matrix between the fibromyalgia condition and the healthy condition. And then what you do is for all the correlations in this row, which is gene 1, you add all these values together. So I would add 1 to the value in orange, to the value in green, to all these values. I would add them all together and get their sum, because the sum would be how the sum of all the correlations between all the genes as they relate to gene Y tell you how impactful gene 1 is in terms of correlation in the cell. And so you would sum all of these up for gene 2 and get a sum here, all these up for gene 3 and get a sum here. So you'd get all of these sums of the correlations between the different pairs of genes in each condition, fibromyalgia versus healthy. And then if you're following what you would do that for the fibromyalgia state and the healthy state. And then what you would do is you would take the values in the sums between the two states and you would subtract them. So let's say that that first sum in blue was, 127. All right. And the sum in blue for the healthy condition was 50 or something like that. So obviously the difference here is going to be 77. Okay, so you would subtract the two. And you would do this for every row of your matrix and get all the differences in sums the whole way down. And I know it's a little bit hard to follow, and I'm going quick here, but the differences in sums tells you how the genes, how each gene's relationship to every other gene has changed between the healthy state and the disease state. This difference in sums, once again, tells you how the relationship of gene 1 has changed between fibromyalgia and the healthy disease state. And so we're looking for a difference in sums that's greatest, which tells you that's the gene whose relationship to every other gene has most greatly changed between the disease state and the healthy state. And I'll put some of these ideas in your notes in just a moment. Okay. And I know this is kind of hard, but are you with me? All right. Okay. So, clearly, okay, Hayden, if it's that easy, then why isn't there a cure? Because we're doing, it's a good question, Hayden, because we're doing math to narrow down the possible genes that we have to biologically investigate, okay? And let me point something out. Just because we know what gene is or isn't causing a problem doesn't mean we can actually get a cure for it. For example, muscular dystrophy. Muscular dystrophy is a problem with the dystrophin gene that causes the dystrophin protein to be abnormal. So people that suffer from this, their muscles deteriorate, and in some very bad cases, their heart muscle deteriorates so bad that their heart can't work anymore and they pass away. Well, we have known about muscular dystrophy for probably 60 years. And we have known for at least 30 to 40 of those years what the problem gene is. But even though we know what the problem gene is, we don't actually have the technology to do anything about it. Okay? So please understand that identifying the cause of the disease is only half the battle. Then we need a technology that allows us to correct the defect in the gene, and that we don't quite have yet. So even though methods like this help us to identify the actual driver of the disease, it doesn't guarantee that we can actually do anything about it. And that's just the reality of the world we live in. Okay. Other questions? Okay, so if you do that for the fibromyalgia data set and you start doing this differential correlation analysis, okay, and I'm going to switch over here, what you will start to see is certain genes start to pop up to the top of the list. They start to bubble up to the surface of the water, if as a figure of speech. And so let me show you what bubbles up to the top. This gene here bubbles up to the top, HLAF, major histocompatibility complex. So folks, if you know the answer to my question, type it into the chat window. what major system of the body does HLA suggest is impacting this disease? Okay. So major histocompatibility complex. What body system seems to be implicated by this result? I know you folks know it. Surely you've had some basic human bio. Major histocompatibility? What is that? Okay, I guess not. All right, well, folks, it is your immune system, right? The major histocompatibility complex is a whole group of genes that are involved in a lot of different immune system processes. So what I'm suggesting is that by doing this analysis, you start to get driving genes highlighted at the top of your list, such as HLA. And if you know what that gene does, you start to narrow down what the problem really is. And so, long story short, scientists now believe that fibromyalgia is not a disease of the muscles where typically you feel sensitivity, but it's actually a disease of the immune system that is very likely involved in some kind of inappropriate inflammatory process in the nervous system. So that when somebody touches you and gives you a hug, your nervous system overreacts to it because the nervous system is inflamed and you feel an unusual amount of pain because of that. So current research is heavily focused on the immune system because computational processes like I'm trying to show you have started to highlight the possibility that immune system genes are heavily involved, potentially as drivers in this disease state. Okay, questions? Okay, so I know this is a lot, and I don't expect you to master all of this. And obviously, if we had another semester, we could do more interesting things. But I'm trying to just get you to see a little bit about how measuring the expression of genes and using those fancy programming skills that you've all started to develop can be used to narrow down which genes are implicated in actually creating a disease. And then, of course, there's much additional hard work to do once you identify the drivers. So I'm going to put some more thoughts that kind of summarize what I've said here into your notes, okay? So the basic idea here for doing this correlation coefficient would be as follows. You calculate the correlation between each gene and every other gene in the same condition, okay, such as fibromyalgia. Okay. And then do the same thing for each gene in the second condition, healthy. Within each condition, you total together, you sum together the correlations for each gene and every other gene. Okay, this provides a quantitative measure of how strong each gene is related to every other gene. Okay. Once you do that, you next subtract the sum totals of correlation for each gene between conditions, between fibromyalgia and health, to obtain a measure of differential correlation, to obtain a measure of differential correlation for each gene. This tells you how much each gene's relationship between the other genes has changed between the disease state and healthy state. Okay. When you do this for the fibro data set, you get genes, bubbling to the top of the list for HLA. Hold on a sec here for, HLA stands for human leukocyte antigen, which is a synonymous term for the major histocompatibility complex. Okay, so maybe that was a point of confusion for you. You get genes bubbling to the top of the list for the major histocompatibility complex, MHC, which implicates the immune system as a driver of fibromyalgia. Okay. Folks, I am not going to ask you to do a calculation like this on your final exam. As I've said, I'm taking a little bit of indulgence with you here at the end of the semester, trying to show you some things that I think are neat, interesting, hopefully allow you to understand some things that maybe you don't yet, but I'm not going to beat you up with this because clearly, you know, you're only going to have 2 hours to take the test next week. But I hope that you're getting a rough idea of what I'm trying to communicate here. Okay? So, questions right now. Right, this is a lot to take in. So sometimes you don't even know what questions to ask because it's kind of like overwhelming when you first hear things like this. And I understand that. So you might have to process it for a while. You know, we as humans have to process things for a while before we can even develop a question. Okay. So, okay. So Hayden, let me come off your question. Okay, well, what next? Let's say that we have identified the driver of the disease through these computations. Okay, wonderful, but what next? Okay, number 9, okay, because remember our overarching topic here is a practical example workflow, transcriptome example. How do we use our fancy technology to do something meaningful with it? Well, hopefully, if you're getting the rough idea here, you see that we use the microarray technology to measure gene expression, and then we use fancy programming in Mathematica or another language that you enjoy to do the calculations to get differentially correlation information, which then begins to prioritize which genes are likely the bus driver, likely driving the disease state. Okay, I mean, granted, everything I'm saying here technically is very challenging. You don't just, you know, whip this out in a 2-hour work session and you solved everything. It doesn't ever work like that. Okay. All right. So now what? So now what? You take the genes that are mathematically predicted as being drivers and you begin to do real biology experiments in the lab to confirm or deny your mathematical predictions. Okay. All right. So, and this is where I think a lot of folks really don't understand what biology is about, because you could have a computer scientist, a bioinformatician, run some huge data set on their computer overnight and get some interesting lists of genes that predict things, and that was done in 2 days time, and then a biologist has to go in and do 3 or 4 months of work to validate or deny that. I mean, biology is hard, tedious, long work that you can't just accelerate like you can by running a computer program faster. It just doesn't work like that. Okay, so a lot of times people don't really understand the biology, and biologists don't get a lot of respect because they figure it's so slow. Well, it's not easy to do this stuff in the biology lab, right? We're not going to have a coronavirus vaccine next month. It just doesn't work. Okay, so let's say, okay, and I'm watching my time here. Let's say you want to know what genes the protein for a driver. Let's say you want to know what genes the protein from a driver gene binds to in a cell's nucleus. Because if we knew that, then we might know what genes have their expression modified by the driver. In other words, we could identify our what, gang? What could we identify if we knew what a driver protein truly was, and we had a way to determine where along your DNA that driver protein attached? What kind of genes might we identify? In my analogy, what kind of genes might we identify? Okay. I understand this is hard. I'm trying to get you to see is that if we had a protein that was made from a driver gene, and that protein were to bind other sequences of DNA, other genes in your genome, we now might have a way of identifying what genes are the passengers. Okay? All right, so let's say you want to know what genes the protein from a driver gene binds to in a cell's nucleus. Let me ask you a question through the chat window. What experimental technique does chapter 14 talk about that allows you to identify specific DNA sites that a protein binds to? What experimental technique does chapter 14 talk about that allows you to identify specific DNA sites that proteins bind to? Do you know? Okay. All right, folks. So maybe some of you haven't quite caught up with your reading in chapter 14, and I understand there's a lot going on. But what I'm suggesting is chromatin immunoprecipitation. If you look on pages 400 and 401 of your textbook, there's an experimental technique described in figure 9 called chip sequencing. Chromatin immunoprecipitation sequencing. Okay? What I'm going to do is show you a little video on that. You could use chromatin immunoprecipitation precipitation to identify segments of DNA bound by a driver protein. Okay. Okay, gotta learn to spell here. Chromatin immunoprecipitation. Okay, folks, so we're getting a little, a little long in the tooth here, and I'm probably getting a little too theoretical at this point for you, but let me finish this out and we'll close up shop, okay? So if you have your list of what genes you think are causing the disease, you got to do some real biology. And one of the things you might want to know is, well, okay, whatever protein is made from my gene of interest, what other segments of DNA does it potentially bind to in a cell, which might help explain why it does what it does. To achieve that, to get that information, you would use a technique called chromatin immunoprecipitation. So let me show you a little video here about the basic idea behind this technique. Here we go.
Chromatin immunoprecipitation, or CHIP for short, is a technique used to determine specific interactions between DNA and a protein, such as a transcription factor. Knowing exactly what sequence on the DNA a protein binds to is useful information that helps scientists determine the function of that protein. Chromatin is the form of DNA that is found in the nucleus. It consists of DNA wrapped around proteins called histones. This allows the DNA to become compacted and take up less space in the nucleus. For the rest of this video, chromatin will be represented by this line for simplicity. In the cell, many proteins are bound to the chromatin. These proteins frequently bind and unbind the DNA, making it difficult to determine interactions. In order to solve this dilemma, first the cell and nuclear membranes are lysed, or degraded, allowing access to the chromatin. Next, a cross-linking agent, such as formaldehyde, is used. This acts like a glue, keeping the proteins bound to the chromatin. Once the cross-linking is complete, the chromatin must be cut into smaller fragments, between 300 and 1,000 base pairs in length. The methods used to analyze the DNA after CHIP will need shorter sequence lengths. Now, there are numerous DNA fragments with different proteins attached. However, in order to determine the DNA binding location for a particular protein, that protein and its bound DNA segment must be separated from the others. This is where the immunoprecipitation part of CHIP comes in. Antibodies that bind only to the protein of interest are added to the solution. These antibodies are in turn attached to dense beads. The resulting complex contains chromatin, a DNA-associated protein, an antibody, and a bead all bound together. The solution is then centrifuged, causing the dense beads and attached compounds to clump and precipitate out of solution. The remaining solution can then be removed, leaving only the protein of interest and associated complex. The final step involves separating out the components to isolate DNA so that the specific binding location of the protein of interest can be determined. The precipitate is heated, undoing the cross-linking glue and breaking the binds between the DNA and associated proteins. This includes both the protein of interest and histones in the chromatin. Once the DNA has been separated, the remaining proteins and beads are precipitated out with organic solvents and removed through another round of centrifugation. This isolates the DNA and completes the process of chromatin immunoprecipitation. Now that the DNA segment binding to the protein of interest has been purified, it can be resuspended and analyzed with a variety of methods. PCR, microarrays, and next-gen sequencing can be used to determine the location of the fragment relative to a gene or location within the genome, as well as the exact nucleotide sequence of the fragment.
Okay, folks. So, I wish we had the luxury of more time together, and honestly, I wish we had the luxury that we could have done more actual real lab work together the remainder of the semester, it would make some of this a little bit easier to understand because you'd have more of that real world experience. But as a first approximation, do you have any questions about that chromatin immunoprecipitation? Did the video make sense what the technique was trying to do? Are you, if you're passed out on your floor, please type yes. Wow, must be bad if you're not even typing yes. Okay. All right. Okay, folks. So let's wrap this up here pretty quickly, okay? Because as I said, I'm kind of indulging myself right now to try to get you to understand some things. But the take-home message is that we do now have the technology to measure the expression of genes in cells, compare them, use some mathematically and computationally sophisticated techniques to get a pretty good guess of what the driver genes for a disease are. And then you have to go into the biology lab and do real work on those predictions to see if you can validate them or invalidate them. Okay, so let's say that the chromatin immunoprecipitation that I'm talking about here was successful in identifying a gene that might also be promoting a disease phenotype, whether it be a rash or whatever it might be. And we want to actually treat a patient by knocking the expression of that gene out. Well, there's a number of ways that we can do it, okay? Okay. Let's say we have determined that a specific gene really is the cause of a disease, and we want to disrupt that gene's expression to relieve the disease symptoms. Well, there's a number of ways we could do this. We could use RNAi, right? We've talked about that. We could use RNA interference. Now, RNA interference is going to be a disease treatment because you don't literally alter the broken gene, you simply suppress its expression. Okay, so this would be a disease treatment, not a cure. Okay, so when we treat a diabetic patient with insulin, when that patient takes an injection of insulin, that is a treatment, it is not a cure. If we were going to have a cure for a diabetic state, we would have to go in and repair the deficiencies in the pancreatic cells that make the insulin. So RNAi, as interesting and cool as it really is, will always remain a disease treatment, not a cure. But there is a possibility of something that I'm sure you've heard on the news and read about, and which we're not going to have in any significant depth here in our course, but we could use something called CRISPR, at least in theory. And I have to say it that way because in theory I can fly to the moon by flapping my arms, but that doesn't work, right? So the devil is always in the details in terms of how do we really make this work. And I think we're going to see some success. I think you are going to see some success in your lifetime, but unfortunately you're also going to see some failures. That's just how it is. Okay, we could use CRISPR. Okay, CRISPR, when it works properly, will be a true cure. Okay, it will be a true cure here. And so rather than trying to walk you through something that's very complicated in 15 minutes, what I'm going to do is show you a quick little video on it so that you get an overview of what CRISPR is all about. Okay, so let's do that. Let me show you a quick video on CRISPR. Okay, and we're going to use this one, CRISPR Gene Editing and Beyond. Here we go.
The CRISPR-Cas9 system is a tool for cutting DNA at a specifically targeted location. The technique has already revolutionized gene editing, but scientists are always looking for new possibilities. So what else can CRISPR do? Since being discovered in a bacterial immune system, CRISPR-Cas9 has been adapted into a powerful tool for genomic research. There are two components to the system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers are deactivating one or both of Cas9's cutting domains and fusing new enzymes onto the protein. Cas9 can then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene, or introduce a stop codon at a specific place. But it's not all about gene editing. Several labs have been working on ways to use CRISPR to promote gene transcription. They do this by deactivating Cas9 completely, so it can no longer cut DNA. Instead, transcriptional activators are added to the Cas9 by either fusing them directly or via a string of peptides. Alternatively, the activators can be recruited to the guide RNA instead. These activators recruit the cell's transcription machinery, bringing RNA polymerase and other factors to the target, and increasing transcription of that gene. The same principle applies to gene silencing. A CRAB domain fused to the Cas9 inactivates transcription by recruiting more factors that physically block the gene. A more outside-the-box idea for using CRISPR is to attach fluorescent proteins to the complex, so you can see where particular DNA sequences are found in the cell. This could be useful for things like visualizing the 3D architecture of the genome, or to paint an entire chromosome and follow its position in the nucleus. CRISPR has already changed the face of research, but these new ideas show that what's been achieved so far could just be the tip of the iceberg when it comes to CRISPR's potential. Whatever comes next, it seems the CRISPR revolution is far from over.
Okay, so, that's a beautiful video talking about the potential of CRISPR. And it is amazing. It is an amazing technology. Okay, I don't want to undersell it. It's an amazing technology. And it does open some new doors for us in terms of how we can potentially treat disease. But, it's not the panacea that your average news report would want you to see, because, CRISPR does have some off-targeting effects. In other words, it binds to things that it should not be binding to. And so one of the problems scientists are having to do is try to genetically engineer the CRISPR enzyme so that it is more precise at what it is supposed to modify and doesn't change things it should not be modifying. So, there's still some considerable work to be done here. but there is real reason to be hopeful. So, folks, do you have any questions right now? Okay, so rather than try to cram other complicated things in 10 minutes, what I'm going to do is stop here and ask you if you currently have any questions about any content that you're worried about for the final exam. Is there anything right now that I can help you with? Do you have any questions about content? Any questions about how we're going to finish up the course on next Tuesday with your final exam? Okay, I'm not hearing anything. So, folks, we're going to close up here at this time. And, you know, this is the last time that we will be together online. And so I just wanted to take a quick few seconds to thank you for your continued efforts and dedication to the course. You know, nobody could have predicted that halfway through the semester that we wouldn't actually be seeing each other anymore. And with all the disruptions in our daily lives and personal schedules, it's been a challenge for everybody to continue forward. So thank you for doing that. I want to ask you to give me a good few days focused effort. You got to finish that homework that's due, right, and get that to me. But when you take that exam next week and you hit the submit button, you can be confident that the semester is over. So thank you for all your hard work. And I do wish you well, you know, in the future, wherever your career takes you. You know, I encourage you to, you know, get a hold of me and let me know how you're doing, because I do appreciate hearing from former students to share in your success. So thank you very much. And folks, I will be in touch through email primarily next week to let you know about any news or things that you have to know about. Otherwise, perhaps I will see you this fall on campus if you're still taking a course. So good luck, everyone, okay? Be safe out there and take care of yourself. See you later.